I'm delighted to be here to introduce the final speaker in our series for the Broad MIT series in chemical biology. We've had a fantastic slate of speakers and a highlight today as well. I want to begin by thanking our sponsors from industry. You can see them listed here. And also, of course, Ed Skolnick, who's made the entire program possible for us. So thank you very much. We really appreciate it. Our speaker today is Jonathan Abraham. Jonathan is a really fantastic scientist in the virology and structural biology space. Jonathan got his start actually at Harvard University, where he earned a bachelor's degree in 2005, and worked with Stephen Harrison there, and actually continued to work with Stephen Harrison as he pursued his MD PhD, earning that in 2012. Following that, he had a term as an intern and resident at BWH, and then was a clinical and research fellow at Brigham and Women's. Really doing beautiful structural biology and virology and a bunch of other great work around immunology all along the way. Jonathan started as an assistant professor at Harvard Med in 2017, was promoted to associate in 2022. And he has also, really impressively, he's also a practicing physician, right? And so I really admire the ability to do all of that. He's got a long list of awards here, all the awards that you dream of getting. So he's won all of the different Burroughs Wellcome Fund awards, from early in the career for physicians, scientists, up through, he's currently one of their investigators in the pathogenesis of infectious disease. He won the NIH Director's Early Independence Award, the Smith Family Foundation Odyssey Award, many other awards here. As I said, his lab is interested in molecular virology, immunology, structural biology of viruses, how they adapt or how they interact with receptors on host cells, how they're neutralized by antibodies. He's interested in the viral polymerases and how they replicate viral genomes. And sort of the big picture vision here is eventually not just to get mechanistic understanding, but also find new avenues to treatments and vaccines and things like that. So it's a very important research area. Jonathan's been extremely successful and made major contributions throughout. You're going to hear about them today. And I should mention that as of last year, he's also a HHMI investigator, which is a fantastic thing to be, I think it's always a fantastic thing to be, but especially if you're working on infectious disease these days, right? So with that, I'll turn it over to him.
Thank you, Matt, for the wonderful introduction. I'll jump right in. And so here on this, maybe I'll stand over here, it might be easier. On this sort of diagram here, I'm showing you a viral particle with a membrane in gray. And the sort of interesting bit we'll focus on today is the viral spike protein shown in blue. And so these spike proteins that decorate the surface of viral particles, of course, played a critical role of attaching the virus to cellular receptors during the first step of the infectious cycle. And so after this binding event, the virus can be endocytosed into, for the viruses we study, acidic compartments, whereby the exposure of low pH allows the spike protein to undergo these massive conformational changes that culminate in fusion of the viral and host cell membranes. And so in addition to our interest in how viruses can bind receptors, we also study neutralizing antibodies that will bind the spike proteins and prevent receptor engagement. And so one of the bigger picture questions we hope our research will help clarify is how spike proteins can adapt to bind cellular receptors in different hosts while also avoiding immune recognition by molecule like antibodies. And so alphaviruses is the virus I'll tell you about today. These are subdivided into two groups, Old World and New World. This is based on their serological properties as well as their geographic distribution. The Old World viruses typically cause rash and arthritis. An Old World alphavirus you might have heard about is chikungunya virus, while the New World viruses typically cause encephalitis. Sort of well-known example in the New England area is Eastern equine encephalitis virus. And so unfortunately for the New World viruses, including EEE, there are no approved vaccines or treatment options. And these diseases can be quite devastating because some of them, for example, have a case fatality rate that can be as high as 75%. And for those who survive infection, they can be left with permanent neurological sequelae thereafter, including many requiring lifelong institutionalized care. And so why we care about these viruses is because they have this pretty interesting life cycle in nature, whereby they are amplified in these reservoir species, including mosquito vectors in birds. This is called the enzootic cycle. And the epizootic end of the cycle here, which are what some might call spillover events, results in dead-end infection when mosquito bridge vectors feed on viremic birds and then spread them to humans or horses. And what I mean by dead-end hosts is that humans and horses will sustain viremia that's high enough to, say, seed the central nervous system. But the viremia is not prolonged enough or high enough to allow these hosts to serve as fresh sources of virus to then spread to new hosts. So infection ends in these hosts here. And so the spike protein is synthesized initially as a polypeptide precursor. You have a capsid followed by a chaperone called E3, E2, E1, as well as this peptide here called 6K that sits in between. And in gray here, I'm showing you the viral membrane. And so really the business end of the particle is E2 that interacts with receptors, and E1 that undergoes the conformational changes I mentioned will culminate in fusion of the viral and host cell membranes. And so these form heterodimers that assemble then as trimers. So there's a total of about 240 E1, E2 heterodimers forming 80 trimers on the surface of particles. And what you have here is E2 that is most membrane distal, and E1 that sits closest to the viral membrane. Capsids on the interior, and the lipid bilayer is here. So I'm showing you a diagram here that doesn't really faithfully reproduce how the virus is actually organized. It has icosahedral symmetry. And so as will be relevant to the other parts of my presentation, we really wanted to take a system that would recapitulate this proper particle geometry as we try to find receptors for these viruses. And so here I'll tell you about sort of a landmark study done out of WashU and Mike Diamond's lab, where I identified this molecule called MXRA8, a cell adhesion molecule, as a cellular receptor for chikungunya virus. And why this discovery was important is that it allowed us to, for the first time, visualize how receptors can interact with alphaviruses. Before 2018, there was sort of critical knowledge gaps in the field, and these molecules were unknown. And in 2018 really is when the CRISPR-Cas9 screens had been used to find this molecule. And hopefully what you can see here is I'm showing you the spike protein, E1 subunit in gray, E2 subunit in light blue. This is a side view. The membrane would be down here. This is a top view. Hopefully you can see that this MXR8 molecule in pink really hugs a cleft that is formed by adjacent E2, E1 heterodimers on the trimers. And so if you use a system like lentivirus or vesicular stomatitis virus, so particles that don't have the proper geometry and that are pleomorphic, you might miss or not form these properly assembled trimers, and that might not give you as reliable a result. And so with this in mind, as we set out to do our own CRISPR screens back in 2017, we decided to, rather than using VSV or lentiviruses, to build our own replicon-based system for alphaviruses. And so we took the alphavirus genome here that has the non-structural proteins, followed by capsid and the structural polyprotein, including the spike proteins here, and split it into two. And so plasmid 1 here has the non-structural proteins, followed by capsid, 2A self-cleaving peptide, followed by a reporter. And so this reporter could be green fluorescent protein if you wanted to study viral entry using flow cytometry or microscopy. And it could also be a sort of selectable marker we could express on the surface of infected cells, like CD20, a B cell marker, that I'll tell you about in the next slides. And plasmid 2 in this case would allow us to express heterologous spike proteins. So in this case, we could take the spike protein of Eastern equine encephalitis virus. And because a lot of these viruses are BSL-3 pathogens, and this is a single cycle system, it allowed us to work with these particles under BSL-2 conditions in our lab safely. And so the way our screen worked is we sort of transduce our library of guide RNAs into cells. So because we were really focused on finding receptors, we only targeted membrane proteins in the genome with a total of 10 guides per membrane protein, so with high guide redundancy. We do our puromycin selection, and rather than using GFP here as our reporter in the virus, we have CD20. So CD20 will be expressed on the surface of cells here. These are 290 T cells we're using for our screen, and expression of this B cell marker would be abnormal and would allow us to effectively deplete these cells using rounds of magnetic sorting. And I say this magnetic sorting is useful because rather than spending, say, 8 hours on the flow cytometer sorting positive and negative cells with GFP, we could just do this with max beads inside a tissue culture hood and do this quite efficiently. And so we do this for several cycles. 3 was sort of a sweet spot for this virus, and followed by next generation sequencing to identify guides that are enriched. And so really, the first screen we did was a virus called Seminichiforous virus. This is just because it got in really well into the 293 T cells. And when we used Seminichiforous virus, the top hit was VLDLR. So VLDLR stands for the very low density lipoprotein receptor. It is a protein that was known to be a receptor for other viruses, including vesicular stomatitis virus and certain groups of human infecting rhinoviruses. HSP9DB1 is a protein that is a chaperone for PCSK9, which is a master regulator of LDLR-related proteins, so that was reassuring. And NTT3A, the role was unclear when we did the study, but it's involved probably in the stability of these other proteins here through glycosylation. And so if you look at how the receptor is organized, it has a long N-terminal sort of ligand binding domain that has what are called LDLR class A repeats. So each of these repeats is about 40 amino acids in length. The repeats are imperfect repeats to each other, so there's a lot of sequence diversity in the region. And they all tend to coordinate calcium, and the same residues that coordinate calcium are typically residues that bind ligands. And so different family members of the LDLR-related families of proteins will have different numbers of these repeats and repeats of a different identities in terms of sequence. And so when we found this as a receptor for Seminichiforous virus, we also wanted to look if other family members could serve as receptors for the same virus. And when we looked at a protein called ApoER2, which is a protein that, unlike VLDLR, rather than being expressed throughout the body, is particularly enriched in the brain. And that receptor also worked as a receptor for Seminichiforous virus. I should add as an interesting point that when we looked at LDLR, the canonical family member, that we didn't see that serve as a receptor for these viruses. And interestingly enough, VLDLR and ApoER2 are known as renin receptors because they bind to a molecule called renin in the central nervous system that's important for development in the brain, and then later on important for synaptic plasticity. And so now, the experiments we tend to perform to validate receptors, I won't go through the whole list in terms of showing you the data, but I'll just explain quickly. The quickest experiment we can do once we have a hit that we're excited about, of course, is to take knockout cells in this case and show that complementing them with VLDLR cDNA in this case allows us to rescue infection. This is measured by GFP quantification of infected cells with the RVPs. We could show that cells that are resistant to infection can become permissive when we overexpress the receptor. We could show that the antibodies directed against the receptor can be used to block infectivity, that purified components interact. This is usually by purifying virus-like particles and recombinant receptors and mixing them together, approaches using biolayer interferometry or ELISAs. And then receptor binding mediates, receptor itself can mediate binding and internalization. Usually we use light cell confocal microscopy. But these are great projects for graduate students because they have a lot of work to do. And in terms of the physiological relevance, this is perhaps more challenging for us. In that case, you have to sort of have an animal model that you show if genetically deficient for this receptor becomes resistant to infection. An approach we use, which I'll tell you about later, is instead, because VLDLR, at the time we were doing these experiments, we thought it was an essential gene to development of mice. So based on prior literature, what we can do is effectively take a soluble from the receptor, administer it to the mouse, and show that the receptor can serve as a decoy and block infection that way. And this is how we usually demonstrate physiological relevance. So we did this all for Seminichiforous virus. At the same time, also we looked at Eastern equine encephalitis virus and saw that VLDLR and ApoER2 could also be receptors for this particular virus. And so now why we were sort of excited about this in the beginning is really the central question we wanted to better understand is how these arboviruses that infect mosquitoes and humans are able to infect cells from organisms that are separated by hundreds of millions of years of evolutionary history. And so VLDLR, interestingly enough, is also expressed in mosquitoes. There it's called lipofullerin receptor 1. And so we set out to express different orthologs of VLDLR and different orthologs of ApoER2. And so by taking our non-permissive cells and overexpressing each of these receptors, we saw that all the receptor orthologs we tested for VLDLR and ApoER2, including bird for ApoER2 and mosquito for VLDLR, could serve as receptors for Seminichi forest virus. Lars Clark, one of the students working on the project, I challenged him to find a good negative control. He tried C. elegans, which also has a VLDLR ortholog that's important for lipid uptake in the guts. I'm not showing you this, but the C. elegans ortholog also worked. So we never really had a negative control for this particular assay, given the promiscuity with which Seminichi forest virus seemed to be interacting with these receptors. Where we looked at Eastern equine encephalitis virus, however, we actually saw that it could bind VLDLR orthologs for some PCs, but not others. So to us, that was sort of the first sign, perhaps, that even though Seminichi forest virus and Eastern equine were using both VLDLR and ApoER2 as receptors, that the viruses might have differences in which they interact with these receptors, which, as a structural biology lab, is sort of inspires us to go to the next step, which is to determine Cryo-EM structures of the complexes. And so whenever we take on a structural project, we want to first map what might be the minimal receptor binding fragment to complex with our virus-like particles for the either X-ray or Cryo-EM experiments. And so the approach we used here was we took individual LDLR class A repeats and put them on the scaffold of the rest of the receptor and then overexpress these to figure out what might be the minimal receptor binding fragment. Now, our hypothesis going in was that perhaps this is flexible. Maybe the most membrane distal repeat would work as a receptor for these viruses. So maybe this construct might work. But when we did the experiments, actually, we saw that multiple of these LA repeats, LA2, 3, 4, 5, LA2, 3, 5, and 6, could serve as receptors for Seminichi forest virus when overexpressed. So rather than engaging an individual LA repeat, multiple LA repeats could be engaged. This also taught us that perhaps there's something unique about the sequences of each of these repeats that makes some work versus not as receptors. And when we looked at Eastern equine and compared it to Seminichi forest virus, we saw that Eastern equine could actually bind LA1 in addition to binding the other LA repeats I mentioned. So again, a different set of data demonstrating that perhaps Eastern equine and Seminichi forest virus had differences in their spike that allow them to sort of engage these receptors differently. So on to Cryo-EM structures. I should mention that for the students in the lab, it's also very stressful because whenever we find a receptor and publish on it, there's like multiple labs that work very quickly to get the Cryo-EM structures. And so there was multiple structures reported over the time frame we did this work. So we did structures of Seminichi forest virus and Eastern equine encephalitis virus bound to the receptor. Here I'm showing you a top-down view. So the membrane would be like in the back of the sort of screen here, and we would be facing the virus. There's the E1 subunit in gray, E2 subunit in light blue, and the E3 chaperone that was retained on our preparations. And so when we looked at binding of the LA repeats for Seminichi forest virus, rather than being in this cleft that I mentioned where MXRA8 binds for chikungunya virus, it was actually at a site that was distal here, about 40 angstroms away on E1 domain 3 here. And it was so kind of like unexpected that the first time we got the Cryo-EM structure, we actually missed that there was density there. So it took us several runs to be able to actually kind of go back and verify that here. For Eastern equine, it was the canonical site, which again is involving clefts formed by adjacent protomers here. So that one was sort of more straightforward to obtain. And so now, if you can zoom into these structures and look to see how the receptor is interacting with the viral glycoprotein, this is the E1 subunit of Seminichi forest virus. This is LA2 repeat of VLDLR. Here you have the calcium ion that's being coordinated by these acidic residues, and effectively 2 lysines that are conserved in Seminichi forest virus but absent in Eastern equine encephalitis virus that are interacting with the side chains of these acidic residues here on the receptor. So this binding mode is engaging a conserved feature of the receptor because these residues are usually involved in calcium coordination. Now, if you look at Eastern equine encephalitis virus, again, 40 angstroms away on the spike, you see a different site now involving E2, shown in blue, whereby you have a cluster of basic residues, again, interacting with the acidic residues on the receptor. I should mention that we also did experiments to show that if you use EDTA, that disrupts the interaction, presumably because it changes the geometry of the site here when you get rid of the calcium and perhaps partially unfolds the protein. Now, if you look at how Semliki forest virus binds to VLDLR, the LA repeats at the bottom here, E1 subunit at the top. It's very similar to how LDLR interacts with one of its physiological ligands, a receptor-associated protein. So typically what you have is you have multiple of these repeats of RAP in this case, because it also is multi-modular, that present lysines that interact with these acidic residues. And if you look at relin, if you look at lipoproteins as well, these sort of basic residue interactions are quite preserved. So really the virus is mimicking in a way the physiological ligand for these receptors. And so summary for the first part, I've showed you that VLDLR and ApoER2 are cellular receptors for Semliki forest virus and Eastern equine encephalitis virus, that divergent receptor orthologs can support alphavirus entry, and that viral spike proteins can bind sites on the spike protein. Viral spike proteins can bind the receptors using different surfaces that are effectively areas where lysines can serve as the critical nidus for interactions. And so now switching to the next part, which is, I think, where the project got even more exciting for us. We were particularly interested about a virus called Western equine encephalitis virus. So most of you in the room probably haven't heard about this virus, and you probably have heard of triple E. So triple E has been around for over a century now, causing episodic outbreaks of viral encephalitis, anywhere from no cases to about a dozen or dozens of cases per year. I think the largest outbreak in the last 50 years was in 2019. There were a total, I think, of 32 cases in 7 states and several deaths. Western equine encephalitis virus is something quite different. So it used to cause explosive outbreaks of encephalitis in humans and horses dating back to the early 1900s, outbreaks involving thousands of horses, hundreds of human cases. But the virus has kind of gone extinct or submerged in North America. The last outbreak really was, or last case was in 1999, and the virus has kind of disappeared since that time. Even though, when I'm in the hospital, when we order serologic testing for suspected arboviral encephalitis, we still check serology. This virus has not caused a case for over 20 years now. And so we were interested in better understanding what was going on with this virus, and so I've showed you that Eastern equine uses VLDLR and ApoER2. I should add that Venezuelan equine encephalitis virus, another member of this family, was shown to bind a molecule called LDLR-related. So this is almost like a mutation construct we would have generated. If you delete these domains, the receptor still works. So LDLR-related has 3 of these LDLR class A repeats, and LA1 is what binds to Venezuelan equine encephalitis virus. But the receptors for Western equine encephalitis virus was not known. And so at the time we were working on Semliki, we also had done a screen for Western equine encephalitis virus, and rather than landing a LDLR-related molecule, the top hit was protocadherin 10. This is a protocadherin that's expressed throughout the body. It's enriched in neurons where it helps synapses communicate with each other by forming these homophilic dimers in trans. And interestingly enough, this was the first bona fide alphavirus receptor that was not an LDLR-related protein, but actually a protocadherin instead. And so what you have here are these cadherin repeats here. And so taking a step back now, looking at evolution of this virus, luckily enough, there's information for strains all the way from the 1930s to 2005. These are phylogenetically described as lineages A, B1, B2, or B3. Interestingly enough, if you look at these strains over time in infectivity studies in mice, the strains from the 30s and 40s are hypervirulent, but the strains from 2005 are virtually non-pathogenic in animal models. And the CRISPR screen we did actually was focusing on a 1971 strain from Montana We had not gone back in time and looked at older strains or looked at more recent strains. And so the student working in the project, Wan Yu, decided to test these various strains, again in groups A through B1, B2, and B3. And I'm showing you here in pink instances whereby the receptors works versus does not work as a receptor when overexpressed in cells. And interestingly enough, when we looked here, we saw that human protocadherin 10 could be a receptor for all strains we tested, but not for the 2005 strains that are non-pathogenic. This was the case also for mouse PCDH10. This was the case for horse PCDH10. So any mammalian ortholog we looked at could no longer serve as a receptor for these viruses. So we were sort of scratching our heads and asking ourselves, what could this virus be using to get into cells? Because we know it can infect certain cell types. When we looked at the bird ortholog, we actually saw that that one could serve as a receptor. So that sort of solved this piece of the puzzle. But this didn't really start to tell us why the strains from the 30s and 40s were hypervirulent. I didn't mention this, but Eastern equine encephalitis virus is maybe the most virulent member of all New World alphaviruses. And some of the old strains are kind of reminiscent of the virulence of Eastern equine. So when we looked at receptor usage, we found that the old strains of Western equine use the same receptors as well as Eastern equine in this case. So in addition to binding to protocadherin 10, they could bind to VLDLR and ApoER2, and this property was lost by newer strains. And so that, of course, led us to do structures of receptor-bound complexes. In this case, I'm showing you again Semliki forest virus. The first structure we went for is the VLDLR binding mode. Again, it binds to E1 for Semliki. Eastern equine binds the cleft. We knew this time to look everywhere on the spike for binding sites, given that we expected differences. The bounding site is similar, effectively involving the clefts for Western equine for the ancestral strain. But when you look at these side views here, again with E1 in gray and E2 in blue, you can see that rather than binding to one LA repeat, there's two LA repeats that are bound, kind of wrapping around the opposite protomer in this case. So this binding mode involves engagement of protomer 1 here. This binding mode involves engagement of protomer 2 with two of these LA repeats bound. So again, evidence of structural plasticity in how these viruses are interacting with receptors. And just to kind of clarify as well, the key here is that you have these clusters of basic residues that are not conserved in the different viruses that are hopping around. So clusters of basic residues here versus here versus here and here that are influencing the ability to recruit LDLR repeats in the binding mode. And so we also wanted to better understand what was going on with protocadherin 10 binding, why certain strains were able to bind the human receptor versus only being able to bind the avian form. And so we obtained structures of PCDH10 bound to E1, E2 for both ancestral strains as well as the 2005 Imperial County strain, Imperial 181, group B3. And so here I'm showing you sequence alignments. Sort of what jumped out at us was effectively that at leucine 149 in the spike protein, in 2005, you get replacement of that non-polar residue with a polar residue, a glutamine. And effectively, this was at an interface. So by getting structures of first, this is a strain from the 50s bound to human PCDH10. We could see the PCDH10 here in orange, the receptor in light blue, that this hydrophobic residue was forming nice hydrophobic interactions in this case with protocadherin 10, but that this residue became a glutamine in Imperial 181, disrupting these non-polar contacts in this case. So you might ask, how does Imperial 181 still bind protocadherin 10? I'm not showing you this here, but it's because this strain in particular has E2 polymorphisms elsewhere at the interface that compensate for these contacts. And we found out that you need about 4 mutations on the E2 protein to compensate for this loss of non-polar contacts in this case. So this is really sort of a barrier that prevents Imperial 181 from being able to recognize human PCDH10 despite its ability to interact with the avian form of the receptor. And so we also wanted to better understand what extent of mutations in the spike, again, will allow the spike now to bind to protocadherin 10 or to VLDLR if you look at the spike from the Imperial 181 strain. And so by examining the Cryo-EM structures together, we basically found that Imperial 181, while it doesn't bind human, horse, but binds bird PCDH10, it is about one mutation away from being able to bind to the human and horse receptors. And with 2 mutations, it's able to bind to all 3 receptors effectively. And so now, how is this information important? I would say it also can help us design potential antivirals. So what we can do is, I mentioned to you these receptor decoy proteins. So we can take, in this case, the EC1 repeat of PCDH10. These structures show that PCDH10 and VLDLR repeats bind an overlapping site in the cleft. So protocadherin 10 EC1 should block access to both receptors, and we confirm this in vitro. So if you take this molecule and you give it to mice that are challenged with lethal doses of the ancestral strain that binds all receptors, you can see in this case that mice are 100% of mice treated with the protocadherin 10 EC1 FC molecule are actually protected from infection as compared to isotype of PBS controls. And if you do a similar experiment and now you put LA1 and LA2, you block access to PCDH10, and again, we saw significant protection in mice given this VLDLR, LA1, LA2 FC fusion proteins. And so ultimately, what we wanted to have is also a roadmap, a molecular blueprint to understand what polymorphisms in the spike protein mean in terms of viral evolution. A lot of this work we were doing while we were looking at the virus in North America, again, that kind of went extinct and submerged. And when we looked at the sequences, we saw that, as I mentioned here, these ancestral strains have these polymorphisms and basic residues here, E81K, E181K, that allow binding to LDLR class A repeats. And then this leucine at 149 up until 2005 allows these strains to bind to human and horse PCDH10, but this ability is lost here. And then as we were writing up the paper, unfortunately, there was a big outbreak of weave in South America. This was 2023 to 2024, involving Uruguay, Argentina, and Brazil. There were over 2,500 cases in horses, over 200 human cases. And so we were sort of eagerly waiting for the sequences based on what we knew here for these polymorphisms. And when these sequences were released here, we could see that all of these viruses had the L149 polymorphism. And interestingly enough, unlike what happened in North America, where the virus kind of submerged by having this L149Q polymorphism, all of the strains we looked at in South America, dating back to the 30s to 2023 now with the freshest outbreak, have this leucine there and are able to bind to both human and equine forms of protocadherin 10. They did not have the signature polymorphisms for binding to VLDLR repeats, though, thankfully. And so I've showed you that weave binds protocadherin 10 as a receptor on mammalian cells, that ancestral strains also had the ability to bind VLDLR and ApoER2, and that with the Cryo-EM structures in hand now, we can better understand how polymorphisms influence receptor dependencies for these encephalitic alphaviruses. And so on that note, I'd like to thank members of my lab. Wan Yu Lee was a graduate student who did a lot of the work on Eastern equine and Western equine in terms of functional assays. Pan Yang and Xiao Yifan are postdoctoral fellows who did the Cryo-EM work. I should mention this is very sort of a field that moves really fast, so many of the lab members here all work collectively together to get the data out very quickly. And various collaborators, in particular Scott Weaver at UTMB, who runs the World Reference Center for Emerging Viruses and Arboviruses, that did all of our BSL-3 work, and then various sources of funding. On that, I'm happy to take questions.
All right, thank you very much. That was great. Questions from the audience. Come up to the microphones to ask your questions.
All right, I'll use this one. Okay, so you had some data where you had like all 8 that you had an effect, and then sometimes like one by itself didn't do much, but 2 and 4 did, or something like that. From looking at the structures, can you understand why some of them have the effect and then some of them don't have, and if that makes sense?
Yes, let me see if I have a sequence alignment. I think the sequence alignment out, sorry. So we did a lot of sequence alignment, sort of gazing. I think one of the key requirements is that you do need to have that tryptophan there for many of the viruses. Let me see if I can find here. So this, in all the binding modes, this tryptophan in the LA repeat stacks against the aliphatic portion of this lysine here. And so that's a critical requirement. For Eastern equine, you could have a phenylalanine here, and it's tolerated. For some leaky, you can't because of this extra, polar interactions between this aspartate here. So we did that sort of like staring at alignments. We get a sense. So whenever this residue is not conserved, there's issues. And for LDLR-83, the molecule that's a receptor for Venezuelan equine, this residue here, whose backbone participates in calcium coordination but not the side chain, is replaced by a leucine, and that leucine prevents these contacts. So we're finally understanding the sort of structure, function, sequence basis for how these things work, and they don't work in certain scenarios.
Thanks for the great talk. So I think you mentioned briefly about the glycosylations, but I wonder, I know that for SARS-CoV, glycosylation of the spike proteins actually enhance the internalizations of the virus into the human cells.
Yep.
So I wonder, have you looked at how the glycosylations impact this affinity, this binding to receptor? And if so, what's a molecular mechanism, if there's any different receptor involved in that pathway, or if it's, what's a molecular mechanism of increased binding affinity?
Oh, that's a very good question. I sort of neglected to mention glycans. I mean, there's some glycans not as heavily decorated with glycans as SARS-CoV-2 or other viruses that have different architectures in the spike. These viruses that have sort of icosahedral symmetry tend to have, I would say, a surface that's more similar to non-envelope viruses. And so for flaviviruses, we know that glycans there, which kind of have a similar shell in general, they bind to DC sign, and that can participate in infection. For alphaviruses, that's really underexplored. I'm not sure if people have looked at carefully, but that's a great question. I should say that E3, though, the chaperone that we saw in our preparations, other groups that had structures did not have E3, and there they saw additional LA repeats bind when E3 was gone. So I think both glycans as well as whether or not the chaperone remains might influence receptor binding affinities, and those are important questions for vaccine design as well, because you want to sort of expose what's relevant when you make a vaccine.
Can you use part of the receptor to treat the disease to keep it from binding from the endogenous receptor, or that's not practical because it's a brain infection?
Right, so one of these experiments here, and I'll just jump to it because it's easier to show. What people have done, and I'll tell you what people have done and why it's sort of tricky to use in the clinic, is make these types of molecules here where you take the repeat that binds to the virus and you put it on the IgG1 FC constant region. So you can give this, 6 hours before viral challenge. You can give this up to, I think, 24 hours post-viral challenge and prevent seeding of the central nervous system. The problem, though, is if you wait too long, the animals become viremic, the virus seeds the brain, and it's too late. And I think of the patients I might see, for example, in the hospital. In many cases, they're coming in, a week later after having their initial fever and viremia. They're comatose in the ICU, send the serology, it takes you about a week to get the result back. And so by then, it's too late. So these are probably better prophylactic agents. And I think in order to use them as antivirals, we need to have better diagnostics where you diagnose things very early on, and then you treat sort of early. Otherwise, you have to figure out how to engineer this to get it. But it's hard to know, because I think later on, as Washington mentioned, those same patients will have positive serology but negative RNA on the PCR from the cerebrospinal fluid, which tells you it's more likely an immune reaction at this point the virus has been cleared. So there are a lot of challenges of rolling this out as therapeutics, and this is more really to get at the point of physiological relevance.
So I have a question about the WEEV. So you have this substitution, was it L149Q,
Yeah.
that cost it the ability to interact with the protocadherin 10. So what's the, I guess what I was wondering, and I apologize if I missed it, but what's the benefit to the virus for that amino acid substitution to have taken over the population as the virus submerged, I guess, right? I mean, there must, what's going on there?
Right, so our hypothesis is that it's genetic drift. I didn't mention this, but there's yet another receptor for Western equine called MXRA8 of the avian species. So chikungunya uses human MXRA8, but not avian MXRA8. And then Western equine can use the avian form of MXR8, but not the human form. And what happens is that mutation is not at the interface with MXR8. And so that mutation is tolerated fine. So our going hypothesis is that perhaps it was genetic drift, not necessarily an advantage, but the virus lost the ability to bind to protocadherin 10, but as sort of a safeguard could still get into avian cells using avian MXR8. Now, another hypothesis that's not popular in the field is that one of the challenges with what's going on in South America with the recent outbreak is that most likely there was to some extent amplification in horses that were not getting vaccinated, with horses being more prevalent there in sort of agriculture. And so one other model would be perhaps that there's an advantage to having a leucine there to be amplified in horses, not as dead-end hosts, but as amplifying hosts. And that's what's keeping the virus in circulation in South America. They vaccinated all the horses, and then now weave has not come back for a second season. And so it's hard to know. In North America, the horses always get vaccinated for Western, Eastern, and Venezuelan.
Great talk. I'm curious to know, so you mentioned your work with horses and bird. Is there additional value from learning from other species, or is it like diminishing returns, or could you speak more to like any plans about diversifying the host or dead-end species?
That's a great question. I'm sorry, I should add that part to the talk. So as I was reading more about this, I found papers from the 50s describing how Western equine was thought to have, as an additional enzootic host, garter snakes, which are sort of the same snakes I saw growing up in my backyard in Canada. And I'm assuming they're sort of prevalent throughout. And the idea is that birds are actually not viremic long enough to be able to, say, carry the virus from South America to North America. But snakes during the winter can be chronically infected with little consequence. And so they're thought to be these overwintering hosts. So in addition to Western equine being able to use avian PCDH10, it's able to use snake PCDH10 as well. And so there's sort of an extra layer as well. That being said, people are not looking aggressively in snakes to see if the virus is still there. But the same mutation, this L149Q, is tolerated by the snake ortholog as well. We have not looked beyond snakes. But based on sequence, I'm suspecting additional might work, like amphibious. Like I was asking the student one, he was like, so how are the snakes getting, how are the mosquitoes getting the virus from snakes? Apparently they bite the eye. And I was like, okay, let's stop talking about it. So. Any other questions? Oh.
More questions? Going once? All right, let's thank Jonathan for a great talk.